What stops those muscles from contracting is the nuclei in the brainstem. We have the medulla oblongata and we have the pons. The medulla oblongata has these two structures. Okay. The ventral respiratory group and the dorsal respiratory group. One in the front part of the medulla oblongata, one on the back side of the medulla oblongata. That's it. Besides the medulla oblongata, we had the pons. And that is technically referred to as the pontine respiratory center, but we know it as the pneumotaxic center. The pontine respiratory center, or the pneumotaxic center, is what ensures breathing really happens. It ensures a smooth transition from okay, that's what rate and depth of breathing, right? Pneumotaxic center. So this pontine respiratory center, this pneumotaxic center, it ensures that breathing happens. Oh boy. We have a dorsal respiratory group and a ventral respiratory group, again in the medulla oblongata. These are going to be active when you are breathing in. Remember, breathing in, inspiration, for eupnea breathing, is an active and then passive event. It is these two groups of neurons that are communicating with one another to ensure that you breathe in, sending impulses into those diaphragms and external intercostal muscles, and then they are inhibited. Okay. And it's these two sets of nuclei that are going to be communicating with one another to ensure that the diaphragm and the external intercostal muscles engage, and then they're not engaged. Stimulated and then not stimulated. Sending nervous impulses into those muscles, and then no sending of nervous impulse into those muscles. What is the nerve that sends the nerve impulses to the diaphragm? Does anybody remember?
Is it the vagus?
Nope. C345 keeps the diaphragm alive. Phrenic nerve is what supplies the diaphragm. The intercostal muscles are just the intercostal nerves. That one's straightforward. Okay. So the phrenic nerve and the intercostal nerves are how these medullary respiratory center nuclei communicate with the diaphragm and the external intercostals. If you're going to be active, you're going to be exercising or under stress or whatever, excitement, well, then you can see one of these is going to be a little bit more active. And the ventral respiratory group is the one that's a little bit more active when you are forced breathing. Okay. Three nuclei. We have our respiratory center in the brainstem. We have the respiratory center within the lung, or within the pons, sorry. The pontine respiratory center, we know that as the pneumotaxic center. The respiratory center in the medulla oblongata is referred to as the ventral respiratory group and the dorsal respiratory group. These nuclei are involved with sending the nervous impulse down to the diaphragm or to the external intercostals to engage, contract, increasing the space within the thoracic cavity. Once they get to the point where they've completely gone to their expansion, they are inhibited. They are inhibited. And it's the pontine respiratory center that's going to ensure that the medullary respiratory center gets turned on and gets turned off, gets turned on, gets turned off, gets turned on, gets turned off. It's a hell of a lot of work. But it's worth it, I guess, breathing. Okay. So these are the anatomical pieces of anatomy in the brainstem that are controlling your muscles to engage. What else can we control? Well, we can also detect chemicals. We can also detect pressure changes, right? Didn't we see this crap when we talked about the heart? Yes. Breathing has the same stuff. What are we moving into and out of the body? Gases, chemicals, oxygen, carbon dioxide. What changes with activity? Carbon dioxide. What changes with activity? Demand of oxygen. What changes with activity? Hydrogen ions. So we can monitor these levels in the body at a multitude of various different parts, or spots, I guess I should say. And we can affect rate and depth, speed of breathing. Oh my God, we have central chemoreceptors and peripheral chemoreceptors just like we had with the blood in the heart to control blood pressure. Got the same thing here. We have some central chemoreceptors. Okay, and here's pictures here showing the ventral and dorsal respiratory groups. Okay, there's the ventral towards the front. There's the dorsal toward the back. There's the pontine respiratory center right there. You can kind of see there's a bunch of communication going on here. All right. So we have some central chemoreceptors. These central chemoreceptors are monitoring chemical changes within the brain. All the stuff I'm boxing here, all the stuff I'm highlighting here, this is the stuff that is very important to know. Okay. We are going to see here in a little bit what happens to carbon dioxide in blood. What does the brain or central nervous system have that we don't see anywhere else? what is unique in terms of circulation that the brain exhibits, the central nervous system exhibits that we don't see anywhere else?
Are you talking about the cerebrospinal fluid?
Even before that. Is there anything that limits? Does everything in the blood get into the brain?
The blood-brain barrier.
Blood-brain barrier, right? Makes a very tight seal around those capillaries, right? So everything that's in the blood can't necessarily get in there and affect the chemistry of the brain. One of the major things that is within the blood that cannot exert any effect on the brain at all are proteins. There are no proteins in the CSF unless you have some kind of viral infection or bacterial infection. Okay. So we are monitoring pH of the cerebrospinal fluid because of metabolic demand. There's a lot of activity occurring within the brain, yes? Electricity moving from a bunch of different neurons all over the brain, all over the time. We get a pH change because we get an increase in carbon dioxide. It's just a byproduct of metabolism. So we get a buildup of CO2, which causes a pH change. We don't want to get too far out of whack with our pH range, okay? So when pH rises, this is a rise in hydrogen ions. Again, this is going to affect pH. I need to get these excess hydrogen ions taken care of, and the only way that I can do that is by helping to get rid of all of the buildup of carbon dioxide. So as activity increases within the brain, CO2 levels rise within the brain, hydrogen ions rise within the brain, affecting pH. We have central chemoreceptors there within the pons that monitor this activity in the brain. If CO2 levels start to rise, hydrogen ion levels start to rise. I need to get rid of that CO2 and ultimately help balance out the hydrogen ions, which is going to affect rate and depth of breathing. This is our fourth piece of anatomy, chemoreceptors within the brain. I have a fifth piece of anatomy, chemoreceptors outside of the brain. These are peripheral chemoreceptors outside of brain.
So the P in front of the CO2 stands for pressure, not
Partial pressure.
phosphorus or something. Okay.
Partial pressure. The partial pressure of carbon dioxide, the partial pressure of oxygen. Gases exert a force, and that force is pressure. Sorry. Air is the sum force of all of the gases inside of it, and all of the gases inside air exert a pressure, and all of that pressure calculates out to 760 millimeters of mercury. Okay, so the P in front is just partial pressure. Okay, partial pressure, write it here. P equals partial pressure. So the partial pressure of carbon dioxide, the partial pressure of oxygen. Okay. peripheral chemoreceptors, they will respond to really a variety of things. They are primarily, from our viewpoint, going to be looking at changes in hydrogen ions. Just like up in the brain, but not like up in the brain. With CO2 increase comes hydrogen ion increase. Totally happens. Happens in the brain, happens everywhere else. But the one thing that we can also get an increase with hydrogen ions is with beta oxidation, fatty acid metabolism. As you use your fat stores to help make energy, well, we're cleaving bonds between carbon and hydrogen ions. So what happens when you consume a high fatty acid diet, or let's just say you don't do carbs, you're a keto person, okay? You don't consume very many carbs. You eat a lot of fats and you're utilizing your fat stores. What builds up? Hydrogen ions build up. This will affect rate and depth of breathing as well. This doesn't happen in the brain very often or well, okay? It's CO2. That's what we look at up there. As a side note, what's one of the first symptoms associated with the keto diet? Anybody know? Any keto people here? Brain fog. Brain fog because you can't generate enough energy quickly from your only source to make ATP, and that's fat. Takes a while.
It
Takes a long
happens with
while for that to build up.
fasting.
Hmm?
Happens with fasting too.
Oh, well, yeah, but
Yeah.
you're using your fat stores there as well. So that wouldn't be ketoacidosis, but it's intermittent fasting. Even though there's no clinical proof to suggest that intermittent fasting can do any of the thing that it suggests. So if you're an intermittent faster, that's great. Keep it up. Do it. If you're deciding whether you should do it or not, don't. Because it doesn't do anything that they claim it to do. Let's see here. So these are the two major ones. Okay, so these are our 5 pieces of anatomy here: the medulla, the VR, and the DRG, the pontine respiratory center, central chemoreceptor, peripheral chemoreceptors. Are there other ones? Oh my God. Yeah, there are other ones. There's proprioceptor. Well, hold on. Baroreceptors that detect pressure changes within the pleura. Baroreceptors that will help to detect pressure changes within the bronchioles. There are proprioceptors that are involved in the breathing process as you just bend over and tie your shoe. Just bend over right now, tie your shoe. Does breathing change automatically? Yeah, because pressure changes as you bend over. If you inhale something, that's an irritant. Okay, there's an irritant reflex. Okay, and that can cause spasmodic reaction within the muscles there in the thoracic cavity, and that can affect rate and depth of breathing. So there's always a lot. That's what I'm trying to get at. There's always a lot. The 5 little pieces, those are the most important ones, because CO2 and oxygen, they're going to be the major things that we're discussing and talking about. Okay, so that's all I'm asking. We're going to get to these last few slides, finish up this concept of controlling the breathing, okay? And then we'll stop for the day. When it comes to normal breathing, and again, this is eupnea. Okay, so quiet breathing or eupneic breathing. Okay. Inspiration happens because of the ventral respiratory group. Well, the inspiratory group. Okay. These fire. What do I mean by firing? What do I mean by this? Where do they fire? A gunshot?
Action potential.
Yes, ma'am. The action potential gets sent down that phrenic nerve, intercostal nerve, and excites them. Okay. As soon as they get to that spot where they are fully engaged, they are inhibited. I've stopped breathing. I exhale. The muscles engage for a couple seconds. Normal eupneic breathing. So for every breath you have in a minute, 12 to 15 breaths per minute, 12 to 15 breaths per minute. Okay. As you sit there and you're breathing, normal eupneic breathing, couple seconds of engagement by the muscles. If you just sit there and breathe normally and just count, you get the 2 to 3 seconds for normal eupnea breathing. And this is all coming from that ventral respiratory group. It is the ventral respiratory group that's going to be involved here. The ventral respiratory group, excuse me, sends the impulse out via the phrenic nerve or the intercostal nerve to those muscles. Engage them. And then they are inhibited. And it's this constant flow of information between the inspiratory neurons and the inspiratory and expiratory neurons within that ventral respiratory group that help us with normal breathing. They're also going to be involved in forced breathing as well. Okay. But these ventral respiratory group neurons are helping us to breathe. Normal eupnea breathing. What about the dorsal group? The dorsal respiratory group are going to be involved with these 12 to 15 breaths per minute. Okay. Those chemoreceptors that we talked about, they send signals to the dorsal respiratory group. The dorsal respiratory group will send the information to the ventral respiratory group. Okay. All of those chemoreceptors that we just talked about, they synapse up here in the dorsal respiratory group. Again, what are they detecting difference of? CO2 levels, which affects pH in the brain. Hydrogen ion levels, which can be affected by carbon dioxide levels. Multiple number of other things as well, fatty acid oxidation. These will relay to the ventral respiratory group. Okay. Chemoreceptors from the brain, peripheral receptors from the body. Come up here and synapse on that dorsal respiratory group. These are my chemoreceptors. I'm going to put C for chemo. Their input comes into the dorsal respiratory group. The dorsal respiratory group's like, ah, crap. We're getting a buildup of stuff here. Carbon dioxide, hydrogen ions. We got to pick up the pace. Okay. The dorsal respiratory group yells at the ventral group. This is what triggers a change. This is what triggers a change. We can affect the rate, and we can affect the depth. Breathing increases when you get an increase in concentration of hydrogen ions in the CSF. This is basically equating to greater increase of carbon dioxide. Got to get it out. If we get an increase in blood hydrogen ion concentrations, which again is a change in the partial pressure of CO2. Your ventilation is going to increase. You're sitting down right now. What if you guys get up and walk across the room? Tell me what's happening. What's happening to the muscles? What are the muscles doing?
They're becoming more active.
They're becoming more active. What do they need then if they're becoming more active?
More oxygen.
They need more oxygen. Where are they getting that oxygen from? From the atmosphere. If they are becoming more active, that means they're becoming more metabolically active. What's a byproduct of metabolism from way back when at the beginning of the semester? Carbon dioxide. We're going to see next time how carbon dioxide is transported. One way is to react it with carbonic anhydrase to get an increase in hydrogen ions and produce what's called bicarbonate. We'll see that a little bit later. But we get a rise in hydrogen ions. Well, I got to get rid of that CO2. How do I do that? By increasing the rate of breathing. Helping to increase that rate of breathing helps to get blood flow into that lung faster, helps to get CO2 out, helps us to get levels of CO2 back down to normal. Everything's the exact opposite of an increase in hydrogen ions concentration in the CSF, decreased levels of CO2 levels within the brain, decreased levels of hydrogen ions or carbon dioxide levels. It would decrease. If you're sitting around like a bump on a log, you're not metabolically active, there's not a huge demand for oxygen, all we're doing is maintaining the status quo. That's it. Okay. We talk about CO2. Oxygen is just as important, guys. If I don't have enough oxygen, I mean, I can't produce enough energy, right? To do stuff. So you're going to be forced to stop. But what is constantly occurring? What is constantly occurring? What is constantly happening here? Metabolism. To sit here and do nothing requires a heck of a lot of energy. So CO2 is always going to be produced. It's a byproduct of metabolism. So CO2 is always going to be there. So when it comes to affecting breathing patterns, the buildup of carbon dioxide is going to be the most important stimulus to affect rate and depth of breathing. Okay. So this stuff right here that I have boxed, that's what you should know. I'm not going to ask dropping 5 millimeters of mercury, okay, by raising blood concentrations of 5 millimeters of mercury, it doubles up the heart rate. I'm not going to ask that, okay? I'm not going to ask that. It is not underlined. It is not highlighted. So I am not going to ask that. Okay. Blood oxygen levels. It's not as you can kind of see there, it's not a regulator of breathing. Oxygen levels have to really plummet for oxygen to be a major effector of breathing. And you see that now. I mean, if you come in with a blood ox of a certain level when it comes to COVID, they're only worried about if it drops below, I can't even remember now, the blood ox levels, okay? Because blood oxygenation it's easy to maintain, relatively speaking, unless you get COVID, then it's not. Okay, and it's easy. It's not important because the receptor is there within the lungs. I mean, there's nothing really to interfere with oxygen easily getting absorbed into the blood there in the alveoli, unless COVID gets in there and affects some of the cells within the lungs, which can basically help form a barrier within the lungs that really prevents oxygenation from occurring. So that's kind of the bare bones about COVID and whatnot when it comes to your lungs. Okay, I'm not going to ask about joint. I'm not going to ask about sneeze or cough reflex. What is ultimately shutting down that ventral respiratory group and inhibiting it? It's baroreceptors. Specifically, it's called the Herring-Brewer reflex. Okay, so this thing's boxed. Okay. We have baroreceptors within the pleura as the lungs inflate. Okay, how do the lungs inflate? When you increase the space in the thoracic cavity. How does the thoracic cavity space increase? By engaging the external intercostals and the diaphragm with normal breathing. So as those lungs start to expand, the pressure, that pressure change that occurs, okay, as we are expanding in that thoracic cavity, increase in pressure. That is what is sending an inhibitory signal, Okay. To shut off inspiration. Did you all sit there and take a big deep breath with me when I did it? I hope. What happened when you got to the very end of your most massive inhalation? Probably a little painful. Okay. That is the Herring-Brewer reflex. It prevents an overexpansion of the lungs. It helps to prevent over inhalation. Ultimately, it really helps to maintain normal levels of oxygen and CO2 within the blood. Because if I bring in way too much oxygen, it's excess. It may not be picked up by the blood running around the alveoli. I may not be able to pick it all up. It may affect the unloading of carbon dioxide as well. So it really helps to keep the right amount of air getting into the alveoli. So that's how this ventral respiratory group is inhibited. Okay. It's the baroreceptors that we find within the pleural membrane, also within the bronchioles themselves. Okay. It prevents them from overexpanding. If the bronchioles widen out, what can they bring more in? More air in. And I don't necessarily need to bring all the air in. Okay. Because I may not be able to utilize all the air anyway. So how does the ventral respiratory group get inhibited? By baroreceptors. By baroreceptors. What have I said this is all about? I've said this is all about pressure gradients. Okay? When pressure gradients change, volumes change, pressure gradients change, air moves. There's also something that we've talked about, but I didn't talk about. When we start talking about the lower respiratory tract, as we talked about the trachea to the primary, secondary, tertiary, to the bronchioles, ultimately all the way down to the alveoli, what has changed with all those tubes? They've gotten smaller and smaller and smaller and smaller as they get down deep into the lungs. If a tube goes from this big to this big to this big to that big, what has changed? The space inside the tube, right? And what happens to the force as we make that hole smaller and smaller and smaller? That affects flow. That will affect resistance. You've altered the flow before of water. If you've ever had a hose and played around with it, right? Turn on the water hose, you open it up and it just goes, just comes out. But you put your thumb over that and cover half of that hole. Well, now I have a smaller space. Water comes out of there a hell of a lot faster now. That's also what happens with the lungs. If I decrease the space of those tubes all the way down from the big trachea to the smallest bronchioles into the alveoli, those alveolar ducts, I am decreasing the size of those tubes on the way down. This helps to speed up and focus all that air down into the alveoli. If they were the same size tube at the trachea as the alveolar duct, air would not flow very fast at all. So by making those tubes smaller, I can increase the speed through which air travels through this bronchial tree.
That's something that for some reason I have a blockage on is when
Okay, a lot of people do with this.
I understand the idea of directly related and inversely related, I think, but then when you go to describe a specific scenario, I'm trying to apply it like, so resistance is, I'm trying to remember, in other words, when the lumen of a blood vessel gets smaller, the resistance increases, right? Is that right? It seemed like that part I could understand, but then is saying, is that directly or inversely related? So it's getting smaller, causing an increase. So decrease in size, increase in resistance. That would be inversely related.
Okay. Flow equals pressure. Okay. So they are directly related. Okay. Flow is equal to change of pressure. It's inversely related to this. Okay. Because of where it sits on the other side of the other side of the math equation. If it's on the bottom, it's inversely related. If it's on top, if it's the numerator, they are equal. They are directly related to one another.
Yeah, the math, I mean, I can see, but I'm saying like how to apply it to an actual scenario. Like you can know that it's directly related, but then when you talk about changing something like the size of the
All right, I hit the button.
vessel.
Okay. So
In other words, if directly related would mean an increase in one is an increase in another. So an increase in size would be an increase in something else versus indirectly would mean.
all right. So flow, okay, and let me just make sure I'm saying this right. Okay, so I'll say it again. All right. So flow is going from the trachea down into the alveolar duct, right? Flow is going to increase in speed as we go from one smaller tube to the next. Okay. Okay. As I change the size of the tube, what am I changing?
The resistance.
Well, I think that's what I said, and I apologize. I'm an idiot. As tube size gets smaller, this is volume. Okay. Large volume, low pressure. Little volume, high pressure. So as flow goes from from bigger to smaller, okay, as flow goes from bigger to smaller, you're going to increase the speed of travel. Okay. Because the change in pressure, delta P, delta is change. Okay, delta is change. The change in pressure at the start to the end, volume diminishes, but pressure increases, right?
Yes.
Okay, so that huge change in pressure that occurs helps to have a huge increase in flow or increase in speed. You with me there?
Yes.
Okay.
So that would be an
Direct.
inverse relationship,
That is
because as one gets smaller or one increases,
the change.
the other decreases.
The change. It's the change in pressure. If we go from this tube to this tube, it's not a whole heck of a lot of change in pressure. But if you go at the very beginning of the tube, trachea, to the very end of the bronchial tree, the alveolar duct, that is a huge pressure difference, change. Do I care? Well, I do care. Do I want air to get to the primary bronchi and then stop, or do I want it to get to all the way to the end of the bronchial tree?
To the end.
Right. So when it comes to a change in pressure, okay, as the volume decreases, the pressure increases. So the pressure here may be, let's say, 0. Okay, not a whole, well, it can't be 0 because nothing would move. Pressure of 1 atmosphere, right? 1. There is such a small amount of volume in here, but I am focusing all of that air into that tube. Here, I have a huge amount of pressure here. And if I subtract where I started from where I ended, that delta P is going to be really large, yes? 1 minus on 99 is 98.
Yes.
I'm just throwing out numbers. It's all about the change in pressure. Hold on. Let me give you an actual number. There it is. I'm going to write this down. I'm going to give you a number to visualize. Doing some math in here. Sorry, I'm doing math. All right. So the trachea diameter is 2 centimeters. The alveolar duct is 200 millimeters. Okay. So that's a delta P of like what? 100? That change in volume results in a huge increase in pressure. Centimeters are bigger than millimeters, right? So it's the difference from the beginning of the tube to the end of the tube. Okay. And air flows faster as the difference between these two gets larger.
Okay.
Okay. So those things are directly related. Greater change of pressure, greater flow. Okay.
Yeah, I have a question.
Yes.
From what you explained here, flow is directly related to volume, because when there's an increase in flow, there's also an increase in volume. But when the volume reduces, the pressure increases. But what I don't understand now is, where does resistance, how do you apply resistance in this issue? Where is the resistance actually there? Because I understand flow, understand volume, increase and decrease, and then understand that when this is, the volume is reducing, the pressure is increasing, but I don't know where resistance.
Okay. Where are we going to get a resistance change? Are we going to get a resistance change in the trachea? Yes or no?
Yes.
No. Why would we get a change in the trachea? We have a cartilaginous ring that keeps the tube a constant diameter the whole time. Are we going to get it in the primary bronchi? Yes or no?
No.
No, for the same reason. We have a C-shaped cartilaginous ring. Do we get it in the lobar bronchi? No, not really, because we have still massive cartilaginous plates. Yes, they're a little bit smaller in size compared to a C-shaped cartilaginous ring, but still, they're pretty big. What about in a segmental bronchi? Well, maybe a little bit now, right? Because now we're starting to get those cartilaginous plates to be a lot more thinner in structure. What do I have a lot more of? Smooth muscle. What happens when we get down into the bronchioles? Plates are gone. And when the plates are gone, what do I have there to maintain the openness of that tube? Smooth muscle. So what's going to, and I'm going to mute you here because I'm getting massive feedback. So what do we have now? If I have bronchoconstriction, what is that going to do? That's going to increase the resistance. Okay. What if you have asthma and we get an increase in the production of mucus within the bronchial tube? What if we get an increase of mucus within the lobar bronchi, within the trachea? That's going to be your resistance. What happens if the alveoli collapse? That's going to increase resistance. Okay. What if I lose the elasticity in my lungs because of emphysema? I'm going to have an increase of resistance because my lungs aren't going to be able to expand as easy. That is resistance. And resistance helps to hinder flow. So the biggest influencers of resistance are going to be infection, asthmatic conditions. Those are going to be your two kind of normal, everyday run-of-the-mill effectors of resistance affecting airflow. Increase in mucus increases resistance. Decrease the elasticity of the lungs. I can't expand very easily, so the flow of air into the lungs is greatly diminished. Does that make sense?
Yes, it does a little bit, but my own problem is from my point of view when I just look at it.
You sound really weird. Type it. You sound like a robot. I can't understand anything you're saying, Imelda. I don't know if that's on my end or your end. I apologize. When it comes to asthma, it is an increase in the secretions of the wall. You get an increase in mucus secretion here. This helps to affect airflow. Let's see if she wrote it down. Decrease in compliance. This is loss of elasticity. Okay. All of this will, oh my gosh, all of this will result in a need to do more breathing, more forceful breathing. Okay. I can't breathe in. It's difficult. I can't expand my lungs. So now the only way to get expansion of my lungs is to engage more muscles. That is where that forced breathing comes in. If you have to sit there and constantly force breathe because you cannot breathe normally because you've lost that elasticity, that puts a huge stress and strain on you. And as you can see, it increases the amount of energy that you have to expend by upwards of 25%. That's a huge amount. You get exhausted if you have to breathe like that, forced breathing. So just that normal eupnea breathing is so easy to do. Okay. Look, I just beat you all over the head with this, I'll put it nicely, stuff. Let's stop here. We'll come back. We'll revisit this next week. Okay. We'll revisit this next week. I get it. Everybody gets kicked in the stomach when it comes to this stuff. It happens every semester. I know. I know. Okay. So let it marinate over the weekend. You're not going to look at it again until Tuesday morning anyway. Let it marinate over the weekend. We'll come back here. We'll revisit it again. I'll put some pictures up. Hopefully those pictures can help represent it a little bit better for you. Okay. So this gets people mind humped every year. Who's still here? I'm going to see who's still here. A lot of people have been